Assalamu alaikum everyone, I am Sana Khan, your biology teacher from the Senior Secondary School Girls, and today I am here discussing you one of your class 12 most important topic, that is Megasporogenesis and development of female gametophyte. So before starting the topic, let us discuss what we have done and what we have discussed in this topic, and that is the structure of megasporangium, process of megasporogenesis, and the development of female gametophyte. So let us start our topic, and our first topic is megasporangium or ovule. So before knowing the megasporogenesis, then we have know that where this megasporogenesis takes place, and that megasporogenesis takes place inside the megasporangium. Now what is megasporangium? So you all already know that megasporangium or ovule is the structure, that is the structure which is formed inside the ovarian cavity with the help of placenta. Placenta is a cushion-shaped structure, and from this placenta ovules arises, and that ovule is a specialized plant structure that produces megaspores. So what is the ovule? That ovule is a specialized structure present inside the ovarian cavity, and it is formed the megaspore, and that megaspore is the structure which develops into the female gametophyte, and that one is the last structure after the meiosis which is formed, or after the megasporogenesis which is formed for the fertilization, because egg cell is present inside this female gametophyte. So let us discuss what is the structure of megasporangium and where the megasporogenesis takes place. So we have to discuss this structure clearly with the help of diagram. Now this one is the diagram of the megasporangium or ovule. So that is the structure of a typical anatropous ovule, in which the outer or first stalk-like structure is present, and that stalk-like structure is what? So this one is the stalk-like cylinder structure from which a ovule is attached with the placenta, is called funicle. So what is funicle? Funicle is the structure from which a ovule is attached with the placenta. Is a stalk-like structure. Now after the funicle, there is a junction, or there is a point from which the funicle is attached with the ovular body, and that structure is called hilum. So what is hilum? Hilum is the structure from which the ovular body is attached with the funicle. Clear? Now after the hilum, this one is the hilum. This one is the hilum from which a ovular body is attached with the funicle, and after the hilum, the 1 to 2 protective layer is present. This is called integument, and that integument is a protective layer of the ovule which covers or which protects the inner mass of the ovule, is called nucellus. What is nucellus? So nucellus is the main body of the ovule, which is composed of mass of parenchymatous cells, and these parenchymatous cells are a nutritive cells which provide the nutrition to the developing embryo cell. Clear? So these two protective layers are present at the above to the nucellus, but these nucellus or these integument is not present at the tip of the nucellus, and that tip and that opening is called micropyle. So micropyle is the region where the integument is completely absent, and it's formed a porous or pore-like structure, and that micropyle is important for the entrance of pollen tube during fertilization. So that is what? So this micropyle is a small or tiny gap, a small opening which enters the pollen tube during the fertilization. Now, opposite to the micropyle, the basal portion of the ovule is called chelicum. That lies opposite to the micropyle, and the inner body of the ovule is made up of nucleus, which I have already told you. Nucleus is the main body of the ovule in which, after the megasporogenesis, embryo sacs develop. I hope you all are clear about the structure of the megasporogenesium. Now come to our next topic, that is Megasporogenesis. Now what is Megasporogenesis? So as the word clear its meaning, Megasporogenesis, megaspore, it means mega for large. And large spores are always female gamete, or it is formed inside the female body. So megaspore means megaspore, and you all already know that genesis is used for produce or to synthesize anything. To produce or to synthesize or to form anything is called genesis. So which structure synthesized here? That is megaspore. So megaspore is a diploid. The megaspore synthesized from a diploid megaspore mother cell, and the formation of megaspore from this diploid megaspore mother cell is called megasporogenesis. It is a very important definition. If we define this, that is, megasporogenesis means the formation of a megaspore from the diploid megaspore mother cell, which is also commonly called as MMC shortly. So when a megaspore mother cell undergoes to meiotic division. How it is divided? So it is divided meiotically. That is also called as reduction division. And in the reduction division, when a cell undergoes to divide, then it gives rise to 4 haploid daughter cells. So when a megaspore mother cell divides, then it gives rise to 4 linear haploid megaspores tetrad. And that megaspore tetrad is haploid in nature. What is meaning of diploid and haploid? So if a cell having 2 sets of genome, then that cell is called diploid. But if a cell having single set of genome, then that cell is called haploid. So the megaspore mother cell divides meiotically or by reduction division, and it's produced or form 4 linear megaspores. Out of these 4 megaspores, 3 megaspores become degenerate. Out of these 4, 3 megaspores become degenerate. And only 1 megaspore is functional. And that functional megaspore develop into the female gametophyte or embryo sac. So I hope you all are clear about the megasporogenesis. So megasporogenesis is the process or formation of megaspore from a megaspore mother cell through meiosis, and by this it gives rise to 4 megaspores, out of which 3 megaspores become degenerate and only 1 megaspore is functional, which develop into the female gametophyte. Now more clear this with the help of diagram also. Okay, let us discuss this megasporogenesis with the help of diagram. And this diagram shows the process of megasporogenesis. So this is the part of the nucleus. One of the nuclear cell differentiate and specialized, and it's behaved like a megaspore mother cell. But this one is the one of the nuclear cell which differentiate and specialize and behave like a megaspore mother cell which is diploid in nature, and that megaspore mother cell undergoes to meiotic division. You already know that meiosis takes place in two stages. First is called meiosis first and then meiosis second. So first, this megaspore mother cell undergoes to first meiotic division and give rise to a megaspore dyad. And after that, second meiotic division, this megaspore mother cell forms a 4 linear megaspore tetrad. Now from these 4 linear megaspore tetrad, this one is the 4 linear megaspore tetrad. 3 of them at the microvilal end is degenerate. Out of these 4, 3 megaspore become degenerate. Become degenerate and only 1 megaspore is functional. This one is the functional megaspore which develop into the female gametophyte. That one is the last mature structure of the female gametophyte. Now how this megaspore which is functional is developed into the female gametophyte, let us discuss about this. So this functional megaspore undergoes to 3 successive or sequential free nuclear division. These are the process which shows 3 sequential nuclear division. First, this one is the functional megaspore, then it's come to the first nuclear division and it's formed 2 daughter nuclei which moves towards the opposite pole, and then these 2 daughter nuclei again undergoes to the second nuclear division. Again it forms 2 another nuclei, then finally it's formed undergoes to third nuclear division, and finally it's formed 8 nucleated cell. And these nuclear divisions are the free nuclear division. Later on, what is the free nuclear division? It means stereokinesis does not takes place or is not followed by cytokinesis. Later on, these 8 nuclei are covered by cell wall, and finally it's formed a 7-cell structure. This 7-cell structure is called the mature embryo cell. So that one is the structure of mature embryo cell. And how this embryo cell is formed? So that embryo cell is formed from the functional megaspore, and that functional megaspore develops through the 3 sequential free nuclear division, and finally it's formed a 8 nucleated cell, and later on when the cell wall is formed and it differentiates into a 7-cell structure and finally forms a embryo sac or female gametophyte. So during this process only 1 megaspore is functional. That's why this is also called as monosporic development pattern of embryo sac or female gametophyte. Why it is called monosporic? Because in this case only 1 megaspore is functional to develop into the female gametophyte. Now come to the next. What is the female gametophyte? So you all already know about the structure of how the female gametophyte is developed. Clear? So that female gametophyte is developed from the 1 functional megaspore that is developed from a functional megaspore, and that functional megaspore forms first 8 nucleated embryo cell. How it is formed 8 nucleated embryo cell? So with the help of 3 sequential nuclear division. After the 3 sequential nuclear division, it's formed 8 nucleated embryo sac, and later on when the cell wall is formed, then it's formed a 7-celled stage. Like that. So when the cell wall formed later on to produce 7-celled stage, now what are these 7 cells? So the 3 cells which are formed at the chelicil end. At the chelicil end of the megasporangium is called antipodal cell. The 3 cells which are formed at the chelicil end is called antipodal cells, and these cells are haploid in nature, having single set of genome. Then opposite to this, the 3 cells which are formed at the micropylar end of the, that is formed at the micropylar end of the Megasporogenesis, that is called egg apparatus. These 3 cells are called egg apparatus. These are 3 egg apparatus. And out of these 3 egg apparatus, one central cell is egg cell, which is a single fertile cell of the embryo sac or female gametophyte. So this one is the single central egg cell, which is the fertile cell of the female gametophyte, and outer these 2 cells are called synergids. These are 2 synergids. Now where is the 7th cell? These 3 which are present at the chelicil end is the antipodal cells. Another 3 which are present or formed at the micropylar end is called egg apparatus, and in the egg apparatus the central cell is the egg cell, which is a single fertile cell of the embryo sac. Now another 2 outer cells are the synergid cells of the egg apparatus. Now where is the 7th cell? So the 7th cell is this one, is the large cell of this, the large central cell of this embryo sac. This one is the central cell of the embryo sac having 2 polar nuclei, is the 7th cell of the embryo sac. And this type of embryo sac is called age-nucleated 7th cell type of embryo sac. Let me more clear this with the help of diagram. This diagram shows the actual mature structure of the embryo sac or female gametophyte. So that we already discussed about this female gametophyte. Again clear this structure. This is the female gametophyte or embryo sac, in which 3 cells which are formed at the chelicil end, I have already told you, is called antipodals. And these antipodals are what? So they are haploid cells. These are haploid cells. Again, the 3 cells which are formed at the micropylar end is called egg apparatus. These 3 are called egg apparatus. And their egg apparatus having 2 different cells. A center cell is called egg, and this egg is a single fertile cell of the embryo sac, which participate in the fertilization. And 2 outer cells are what? So these cells are the synergid, and that synergids having filiform apparatus, that is a finger-like projection. So what is this filiform apparatus? It is a finger-like projection which is present at the base of the synergid, which guide the entry of the pollen tube to the egg cell. These filiform apparatus is a structure which helps guiding the entry of the pollen tube towards the egg cell. Clear? Now what is the 7th cell? So that is the main central cell. The 7th cell is the main central cell with two polar nuclei, and we have, due to the presence of these two polar nuclei, that central cell is the single cell, which is the diploid cell of the embryo cell. So I hope you are clear about the structure of the embryo cell. What is the structure of the embryo cell? So that is a 7-cell, 8-nucleated structure in which 3 cells are antipodals, another 3 are egg apparatus, in which a single egg cell is a central cell, another 2 are the synergids, and a central large cell having with 2 polar nuclei, which later on converted into the secondary nucleus is the central cell. Finally, this 7-cell, 8-nucleated structure is also called as a common polygonal type of embryo sac or female gametophyte, which participate in the further process called fertilization. Now let more clear this with the help of a final diagram with the conclusion. So our today's topic is megasporogenesis and the development of female gametophyte. So how the megaspore forms? That megaspore is formed from the megaspore mother cell. That megaspore mother cell, with the help of meiosis, undergoes to reduction division, and finally it's formed a 4 linear haploid megaspore, and out of these 4, 3 megaspores become degenerate and only 1 megaspore is functional, and that functional megaspore undergoes to 3 sequential free nuclear mitotic division, and finally it's formed 8 nucleated structure, an 8 nucleated cell, and later on when the cell wall is formed, that 8 nucleated cell forms a 7-cell, 8 nucleated structure, which is the mature embryo sac, a female gametophyte. Just before fertilization, these 7 nuclei present, and out of these 7, 6 nuclei or 6 cells are haploid. Only a single cell which is diploid, that I have already told you, that is the central cell with 2 polar nuclei. I hope you all understand about the megasporogenesis and how the embryo sac of female gametophyte develop inside the megasporangium. Thank you.